Today we're talking about human pedigrees. You may have heard of a pedigree because it's the name of a dog food, but in reality, when it deals with genetics, a pedigree is an illustration to show how certain traits are inherited through families. So we'll go over what a pedigree looks like, how to make a pedigree, and then we will discuss different inherited traits that run specifically in humans. So we're going to get away from Mendel's rules a little bit today. Pedigree analysis, to analyze a pedigree. This means to analyze the inheritance in human families. So it's kind of like a family tree. You take a family tree, you look at the traits that each individual has in that family, and their phenotypes, and from that, you can study human inherited diseases. This allows individuals to infer things concerning the genotypes in their family, or you can look at an entire population and the genotypes that might be in that population. Remember, genotypes are the genes that someone actually possesses. So without going in and taking samples of their DNA and then analyzing that DNA for its molecular structure, you can look at the traits that they have, you can look at their phenotypes, and you can say, okay, well, if this person has this phenotype, then there's only one possible genotype that they could have, and it has to be big A, or little a, or whatever. So it allows predictions concerning the phenotypes, what the individual actually looks like, or its physical characteristics of an offspring inheriting a genetic disease. This has led to an entire occupation called genetic counseling, where individuals deal with families who have possible genetic diseases that they could pass to their offspring. And these genetic counselors then can give information to the families if they are considering having children about how likely it would be for their children to inherit certain genetic diseases. Genetic diseases typically affect a very small number of offspring. And so the laws that we've been dealing with that Mendel created, those ratios are rarely observed, which means the normal 3-to-1 dominant to recessive ratio doesn't usually happen. You don't usually have, in a family of 4, you would not necessarily have 3 individuals with a trait and 1 individual without, or vice versa. So to create a pedigree, you typically have 4 common symbols, and this is what they stand for. A blank square stands for an unaffected male. A blank circle stands for an unaffected female. A shaded square or circle stands for males and females that show that trait or that are affected by that trait. There are some other common symbols that we need to be aware of. A line drawn between a male and a female means that those individuals have mated and had offspring. To show the offspring, you draw a line down from that mating line, and then you spread that line out to indicate the number of offspring that those 2 parents have had. You put the offspring in birth order, so if you have boy, 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 girl, you would see square, 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 circle. If you have fraternal twins, then you have 2 lines coming from the same center point. If you have identical twins, you have those same 2 lines coming from the same center point and a line connecting the 2, which in this case does not mean that they've mated, just that those are identical twins. In some rare cases, you wouldn't know the sex of the child. For example, if it died before birth, if it died, or if it's historically, it was just unknown and you don't have any historical records for that far in the past, then you use a diamond. Again, affected individuals are completely shaded in. If you have a certain trait where the individual can carry the gene for that trait, but they don't actually show that in their phenotype, then you only shade in half of the symbol. This is another way to show the carrier of sex-linked recessive traits. If an individual dies, you kind of cross out their symbol, and so on and so forth. So what I'd like to do is describe the pedigree on the next slide for the following questions. You'll see these questions again on the next slide. Number 1, what disease or trait is being shown? And keep in mind that these questions are on your assignment, so make sure that you're filling in the answers as we go. Number 2, is person 1-1 male or female? 3, has person 2-3 mated? 4, how many children do 1-1 and 1-2 have? 5, which individuals in this family have cystic fibrosis? And 6, compared to the individuals in generation 1, are the individuals in generation 3 children, parents, or grandchildren, or siblings? So let's take a look at the pedigree. This is an example of a pedigree for cystic fibrosis. Just so you are aware, the Roman numerals in the pedigree show the generation. So here's generation 1, here's generation 2, here's generation 3, and all members of the same generation are lined up across in a row. The numbers, the actual numbers, 1, 2, 3, 4, show the birth order of the children. So let's take a look at those questions again. Number 1, what disease or trait is being shown? Number 2, based on what we learned about shapes of symbols, is person 1-1 male or female? Number 3, has person 2-3 mated? So you'd be looking for this kind of line to indicate that that individual has mated. Number 4, how many children do 1-1 and 1-2 have? When you look for children, you look for the line coming down, and then you look for any symbols that fall below that line. But remember, it still has to be connected to that line in order for it to be mated. Or in order for it to show children. Number 5, which individuals in this family have cystic fibrosis? And number 6, compared to the individuals in generation 1, which would be these two parents here, are the individuals in generation 3, which would be this line here, would they be children, parents, grandchildren, or siblings to generation 1? Based on this same pedigree, we can determine a reasonable guess as to what the genotype of the parents and the offspring are. So we can actually tell, based on what we see here, we can guess with pretty distinct certainty what their genes are, what the alleles that they inherited were. And here's how you do that. Assuming that this is a recessive gene and it follows some of Mendel's laws, we know that individual 2-3 and 3-4 must be little a, because they show the recessive trait. Therefore, both parents of these offspring must have been given a little a allele. So that means parent 1-1, 1-2, 2-4, and 2-5 must each have a little a in their genotypes. It's okay if you're not following along. I'm going to show you in the next slide exactly what this looks like. And number 4, since those 4 parents don't have the disease, their other allele must be a capital A. Since they show the dominant trait, they must have at least one dominant allele. And lastly, all the rest of the people in the pedigree could either be homozygous dominant or heterozygous. So let's take a look at what I actually mean. Start with the recessive individuals. We know that if this is a recessive trait, they have to have 2 lowercase alleles. That means they must have gotten one of each of these little a's from their parents. So their parents both have to have at least one lowercase a. However, we also know that their parents do not show the trait, whatever trait it happens to be. So their parents show the dominant trait, which means the other allele that the parents possess must be a dominant allele, which would be represented with a capital letter. That's what we can tell for certain. After that point, the rest of the children from these parents could either have heterozygous or homozygous dominant genotypes because, for example, in parent 2-2, or I'm sorry, in child 2-2, she could have inherited either a capital A from mom and a capital A from dad, giving her a homozygous dominant, or she could have inherited a capital A from one of her parents and a recessive A or a lowercase a from the other parent. So her genotype, as much as we can determine, we only know that it would be either capital A, or capital A lowercase a. And that's true for all of the rest of the individuals. That's as much as we can determine from this pedigree, from what we see. Now it's your turn. What are the genotypes or possible genotypes for each individual in this family? Use the capital B to represent dominant alleles and lowercase b to represent recessive alleles. On your assignment, make sure you label each individual in the pedigree with their generation and birth order number. So Roman numeral 1 would be what genotype. Roman numeral 1-2 would be what genotype, and so on and so forth. If you need a reminder of how to do this, go back a couple slides or rewind the movie. Now there are certain categories of inheritance. Autosomal means that the gene is inherited anywhere on chromosome 1 through 22. Sex-linked, however, means that the trait is inherited either on the X or the Y chromosome. X-linked recessive means that this allele is found on the X chromosome, and it can be found in males or females, because remember, both males and females have at least one X chromosome. A male would have one X and one Y, a female would have two X's. This also means that both males and females can pass this gene onto their offspring. I was wondering why there was a space there. Autosomal recessive or autosomal dominant. These follow the normal Mendel rules, and they get inherited on chromosomes 1 through 22. Back to X-linked recessive. For example, colorblindness and hemophilia are two X-linked recessive traits. You could also have X-linked dominant. These are very uncommon diseases or traits. But this means that the allele is found on the X chromosome, and it could be in male or female. And even if one of the chromosomes has this trait, the offspring will definitely show that in their phenotype. In the X-linked recessive, that means in females, both X chromosomes have to have the recessive trait on it in order for a female to show that. But males, since they only inherit one X chromosome to begin with, if that one X chromosome has the trait on it, then they will show that trait. And Y-linked. This means that the allele is found on the Y chromosome, and since females don't inherit Y chromosomes normally, this trait is only found in males. So let's look at each of these now. Autosomal recessive traits and their pedigrees. This is an example of an autosomal recessive pedigree. It looks fairly normal. The trait is found only rarely. It often skips generations because it can be hidden in heterozygous carriers like we saw earlier. This is the pedigree we saw earlier, where we labeled their genotypes. So the trait can be found in or carried through heterozygous individuals, but it would not be shown. It affects males and females equally. And some possible diseases that are autosomal recessive, meaning found somewhere on chromosome 1 through 22 and follow the recessive rules, are cystic fibrosis, sickle cell anemia, phenylketonuria, which is PKU, and Tay-Sachs disease. If you're interested in any of those, feel free to look them up. Autosomal dominant, on the other hand, has a different look to its pedigree. The trait would be found on the purple individuals in this particular pedigree, not the light blue ones. You can see that the trait if it's an autosomal dominant, is a much more common trait. It's found a lot more frequently. It's found in every generation, meaning it would not skip generations. And affected individuals transmit the trait or give the trait to about half of their children, regardless of the sex. So it can be found in males and females, and it shows up generally in about half. Something to note here. If we look at, for example, apparently this female here mated with two different males. These are the children from the first mate, and these are the children from the second mating. Only the ones that are connected with this line that runs down like that are actual children. If it doesn't have a line that connects to that offspring line, that means that is who that child married or mated with. So the children of these two parents, an affected female and a non-affected male, were boy, girl, 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 boy. So they had 5 children, not more than that. That means their oldest child got married, their second child got married, their third child did not, their fourth child got married, and their fifth child did not. So just a little note on how to read these pedigrees when they start to get big. Some examples of autosomal dominant traits. There are very few autosomal dominant human diseases, but some rare traits have this inherited pattern. Why do you think there are so few autosomal dominant human diseases? Probably because if an individual inherits this dominant trait, it's very possible that individual, that offspring, would not survive to actually be born. So some examples of traits that do follow these rules: achondroplasia. This is a skeletal disorder causing dwarfism. If you've ever seen the show Little People, Big World, I know at least one of the parents says that their dwarfism is caused by achondroplasia, which is why they could pass it on to their children. And Huntington's disease. Huntington's disease affects muscle coordination and brain function. On to the next kind, X-linked recessive pedigrees. So this means the trait is found on the X chromosome. It's very rare in the pedigree. You can see that only 3 individuals in this family have been affected. It skips generations. Nobody in this generation inherited it, even though you know that at least one X from this parent had to have passed on to these individuals. Since it follows the recessive rules, that means you have to have both parents offering an X with the recessive trait on it. Affected fathers do not pass it on to their sons. And males are much more often affected than females, although females can be affected. It shows up more in males because, remember, a male only has one X. So if either the dad or the mom passes on the X with the recessive trait on it, then that would show up in the sons more frequently than the daughters. Females would be carriers. You can pass it from mom to the son. An example of an X-linked recessive disorder is hemophilia, which in history greatly affected the European royalty, starting back with Queen Victoria, who lived in the mid to late 1800s. She passed on hemophilia to her children, to her sons, and her daughters became carriers. So you can see Victoria's son, Leopold, had the disease, where her daughters, Alice and Beatrice, were carriers. And then this moved on so forth down the line. Another X-linked recessive trait is glucose 6-phosphate dehydrogenase deficiency. You don't really need to know that full name. If you're interested, it's a hemolytic disorder that causes jaundice or yellowing of the skin in infants. It's often fatal. and sensitivity to fava beans in adults, meaning they would have an allergic type reaction to fava beans. This is the most common enzyme disorder throughout the world, and it's found especially in those who live or grow up in the Mediterranean. And it may give, on the positive note, it may give individuals resistance to malaria, which is kind of a funny thing, how genetic diseases can have severe symptoms that are often negative, but they can sometimes also give some positive symptoms. Moving on to X-linked dominant pedigrees. These are pedigrees that look like this. The trait is very common in this pedigree. You can see the blue affected individuals show the trait. As you can tell, it's found in both males and females. Fathers will pass it to all of their daughters because if it's on the X chromosome and it's a dominant trait, the daughters have to get that X from their dad. They would also get an X from their mom, but their mom could have two possible choices to give them. So the X-linked, or the X chromosome from their father, if it carries this dominant trait, it wouldn't matter what X their mother gave them. If it's on the X from their father and it's dominant, then it would show up in all the females. Males and females are equally likely to be affected. X-linked dominant diseases are extremely unusual. They are often lethal, as in before birth in males and only seen in females, which is why you don't see it show up quite as often in the males. Here are some examples: skin lesions, X-linked rickets, which means bone lesions. And lastly, thinking about all of these things, pedigree analysis in real life, you have to remember, dominant traits may be very rare in a population. For example, achondroplasia dwarfism is a very rare trait, even though it's dominant. Recessive traits may be very common throughout a population. Alleles may come into the pedigree from two different sources, which allows for a lot of variability. And you have to remember that mutation happens. Sometimes, if a random mutation occurs in someone's DNA, one of these traits can show up where it was never seen before. Often traits are more complex than we think about. They can be affected by environment and other genes. If you have any questions, please feel free to get a hold of me either by message or by email, and hopefully this clears up some things about pedigrees.